Welcome to our channel Mootechie, Microbial Classification Systems. Explore microbial classification with a modern approach. This introduction sets the stage for a clear, engaging look at how scientists categorize the vast world of microorganisms using visuals and straightforward explanations to make complex topics accessible. Now, let's get an overview of microbial classification systems. The purpose is to organize microbial diversity and enable consistent nomenclature. This is crucial because without a common naming system, communication about microbes would be chaotic. Major approaches include phenotypic, genotypic, phylogenetic, and polyphasic. Each offers a different lens through which to view microbial relationships. Taxonomic ranks flow from species to genus to family and on to higher levels, much like a biological filing system. The current trend is a shift toward genome-informed and polyphasic frameworks which combine multiple lines of evidence for greater accuracy. Let's begin with the traditional classification method, the phenotypic approach. This method is based on observable traits. Morphology looks at cell shape like cocci or bacilli, their arrangement and motility features helping to visually group similar-looking organisms. Staining profiles such as gram status, positive or negative, acid-fast staining, and endospore visualization provide quick chemical fingerprints of cell wall structure. Physiology examines metabolism type, the temperature and pH ranges an organism can tolerate, and its oxygen requirements, telling us about its lifestyle. Biochemical tests like API strips, enterotube systems, and specific enzyme assays reveal the metabolic capabilities of a microbe through its chemical reactions. The pros are that it's rapid and low cost, excellent for initial identification. However, a major con is that trait convergence, where unrelated organisms evolve similar traits, can be misleading, making them appear related when they are not. Moving into the molecular age, we have genotypic classification, DNA-based approaches and molecular tools. DNA relatedness uses techniques like DDH, DNA-DNA hybridization, and ANI, average nucleotide identity thresholds, to define species boundaries based on genetic similarity. Marker genes, particularly the 16S rRNA gene, but also GIRB and MLSA, multi-locus sequence analysis, are used as standardized genetic barcodes to determine relatedness. Genome-based classification leverages whole genome sequencing, WGS, where an ANI of 95% or higher is a common benchmark for considering two organisms the same species. Strain typing methods like PFG, MLST, and SNP analysis offer high resolution to distinguish between very closely related variants. The great strength here is the high resolution and objectivity. The primary limit is that it requires significant sequencing resources and computational power, which can be a barrier. This brings us to phylogenetic classification, which focuses on evolutionary relationships and history. It aims to reconstruct the evolutionary history of life by comparing homology, shared ancestry, signals in conserved sequences. Common markers include the 16S rRNA gene for bacteria and archaea, 18S for eukaryotes, or sets of core genome or housekeeping genes which evolve slowly and are good for tracking deep relationships. Methods like maximum likelihood, Bayesian inference, and building distance-based trees use algorithms to infer the most likely branching patterns from genetic data. The outputs are evolutionary trees showing clades, groups sharing a common ancestor, with statistical measures like bootstrap support values indicating confidence in the branches. A major caveat is that phenomena like horizontal gene transfer, HGT, and recombination can blur these evolutionary signals as genes are swapped between unrelated lineages, complicating the tree of life. To overcome the limitations of single approaches, modern science often uses polyphasic taxonomy, an integrated classification framework. It integrates phenotypic, genotypic, and phylogenetic evidence to reach a total consensus on an organism's placement, making the classification more robust. It relies on type strains, standardized reference isolates that serve as the official benchmark for a species, with amended descriptions as new data emerges. The general workflow flows from isolate to phenotypic profile to genetic sequence to comprehensive analysis and finally to circumscribing or formally defining the new taxon. It follows standards set by bodies like the ICSP, the International Committee on Systematics of Prokaryotes, and requires valid publication in a journal like the IJSEM, the International Journal of Systematic and Evolutionary Microbiology. The final outcome is a robust, reproducible, and stable classification system that stands up to scientific scrutiny. Let's now compare these classification systems side by side. Looking at resolution, phenotypic methods offer low to moderate resolution, typically at the genus or species level. Genotypic methods provide high resolution down to the strain level. Polyphasic taxonomy aims for a robust consensus classification. Considering the data needs, phenotypic relies on lab assays and culture media. Genotypic requires sequencing and computational power. Polyphasic needs integrated multi-source data. For typical use cases, phenotypic is common in clinical diagnostics and quick field ID. Genotypic is key for evolutionary studies and disease epidemiology. Polyphasic is the gold standard for new species description and formal taxonomy revision. Each has its limitations. Phenotypic methods can be misled by trait plasticity and convergence. Genotypic approaches must contend with debates over HGT events and where to set similarity thresholds. The polyphasic approach, while comprehensive, can be resource-intensive and complex to execute. The slide's conclusion offers the best practice to synthesize all available data via a polyphasic approach for the most stable and accurate classification possible. In summary, here are the key takeaways and conclusions. Classification structures the immense microbial diversity and standardizes names globally, which is foundational for all biological research. Phenotypic methods are practical and accessible but can be limited in their taxonomic resolution. Genotypic and phylogenetic tools allow us to peer into the DNA to reveal true evolutionary relationships that physical traits might obscure. Polyphasic taxonomy, by combining all evidence, yields the highest consensus and ensures nomenclature stability over time. Ultimately, you should choose methods based on your specific question, like simple identification versus formal taxonomy, the resources available, and the scale of your study. Thank you. Subscribe to Mootechie for more. We hope this tour through microbial classification has helped clarify how we bring order to the microscopic world. Keep exploring, and we look forward to sharing more deep dives with you soon.